Okay, hi everyone. Today, we're going to continue again with what we are calling visual processing. Okay, so basically, we're looking at introduction to the human eye, or to be more precise, we looked at the anatomy of the eye so that we can now look at the physiology of sight or vision. So now what you're seeing on the screen is showing the parts that we looked in the previous 3 videos. So if you have not checked out those videos, please go back to those videos before looking at these slides. Okay. So now we have what we call the fovea centralis, that you already know that they are made up of more cones as compared to the rod cells. So basically the rods and the cone cells, they are the light sensitive type of cells. The rods are responsive to dim light, while the cones are responsive to bright light. So we'll talk about the physiology of how visual processing can occur or can happen as we go on. So now, what you're seeing here, these are known as optic nerves. Then we have what we call the retina, okay? Blind spot, as you can see here. So this blind spot only consists of optic nerves leaving the human eye, so there are no light sensitive cells. So if an image falls to this part, nothing is going to be recorded, okay? Then this is the vitreous humor, as we looked at the other time. And there's also the cornea, there's also aqueous humor and the lens. So the cornea, aqueous humor, the lens, and the vitreous humor help in what we call refraction or bending the light rays as they pass through these structures, so that we can have, or we can achieve, proper accommodation or just accommodation. So now on the far side where you're seeing the cursor, you're going to find that these at the center, okay, we have what we call the fovea, high concentration of cones or photoreceptor, like I said. Then there's a blind spot, as you can see, that is being shown by something very white, which is on the periphery or on the sides away from the fovea. We have what we call the peripheral region, which is predominantly made up of rod cells. So today we're going to be looking basically how can an image be interpreted, how can it basically the rods and the cone cells. So now we have the retina. The retina contains the visual receptors known as the rods and the cones. There are about 6 million cones. Of the rods and the cones varies in different areas of the retina. So you're going to find that as we are starting to move away from the fovea, we are going to have more and more rods as compared to cones. Fovea centralis has mainly what we call the cones. So while proceeding from fovea towards the periphery of the retina, the rods increase and the cone decrease in number. At the periphery of the retina, many rods are present, just like we have been talking about. I'm sure you know. So now, let's look at the rods. So basically, so what you're seeing on the far side of the diagram, we're able to see the cursor. These are just showing the layers that we looked at in the previous video. If you didn't check that out, please go back and look at that. So what you're seeing, this is a choroid. The choroid is a melanoid layer, okay? And then after that, we have got what we call the epithelium layer, or one pigment epithelium. As you can be, both light sensitive cells. So we have got the rods and the cones. So if you look at the tip of the rods and the cone cells, you can be able to differentiate by the structure. This one is looking like a cone is known as the cone cells, and these are known they're known as the rod. So if you can even look at the appearance, you can say this is looking like a cone and that's looking like a rod. Okay, so hence that's where the names come from. So this is known as the photoreceptor layer. Okay, and there's also the number 3, which is the outer limiting membrane. Okay, so number 1, pigment epithelium. Number 2, photoreceptor layer. Then you have with number 3, the outer limiting membrane. Then as you go up, okay, there's number 4, the outer nuclear layer. So here is a part which is consisting about number 2, number 3, number 4. And part of number 5 is consisting of what we call the light sensitive type of cells. So after that, then you know they have got what we call horizontal cells, I've got macrine cells, bipolar cells, etc., etc. So when light comes, it first excites this type of cells, okay? But if anything goes into the choroid is able to absorb because it contains melanin, okay, known as called a melanoid type of tissue. Since you know how it moves, so The first cells that are supposed to be excited are the photoreceptor cells. Then after that, it's passed on to the bipolar cells. After the bipolar cell, then the ganglion cells, then hence it goes to what we call the optic nerves and then transported to the brain for processing. So now let's get back to this. So the outer segment, long and slender in contact with the pigmented epithelial cells, like we have looked at. Discs in rod cells are close structures with rhodopsin. Okay, rhodopsin is a type of substance that is found in rod cells. Okay, so discs in the rod cells are close structures with rhodopsin. So you see the importance of this substance. I mean, this substance known as rhodopsin, which we'll be talking about throughout the discussion about the rods. So now the inner cell, so remember, when you look at this other side of the rod cells and the cone cells. So this is a cone cells, it looks like a cone at the tip. These are rod cells, looks like a rod. So the inner segment, those are the three segments. So there's outer segment, inner segment, and synaptic endings, okay? So the outer segment, that's what you're looking at, which contains rhodopsin, okay? Rhodopsin is contained by the outer segment. Then the inner segment contains organelles with many mitochondria, okay? The inner segment contains organelles with many mitochondria. And you know the reason why we need mitochondria. Mitochondria act as a powerhouse where it produces ATP, as you know, and ATP is the energy currents in cells. Cells cannot live without ATP because they need a form which they can tap energy to as quickly as possible, okay? So there's ATP, there's creatine phosphate, but basically we're talking about ATP, which is very, very common in cells because that's the current, it's more like money in a cell, okay? So without it, a living organism is non-functional. Now let's go to the cell body, okay? So now cell body arises from the inner segment passing to the outer nuclear layer through external limiting membranes, and it forms the cell body that contains the nucleus. Okay, so what you're able to see there is the cell body where there's the nucleus. All right, then we have got the synaptic terminal. Now I always tell my students to say, please study cell physiology and understand and appreciate cell physiology. Okay, so synaptic terminal in the outer plexiform layer, synapsing with dendrites of the bipolar cells and horizontal cells. So these synaptic endings, okay, they can able to form a synapse. A synapse is just basically a junction between two adjacent cells that creates a form of communication, right? Yeah, so if you did well in cell physiology or you know cell physiology, I believe when you just talk about synapses, it just clicks your mind and you know what you're talking about. And it's very important for you to know the foundation of physiology, which is cell physiology, okay? So now it contains a neurotransmitter known as glutamate. So glutamate is found at the nerve or synaptic endings of, at the not nerves, but at the synaptic endings of the rods and the cone cells. So it's this same glutamate which is able to cross over the synapse. Remember, under normal type of cells, under like what's it, neuromuscular junction, we had what we are calling acetylcholine. Okay, so now instead of acetylcholine, we're going to have what we call glutamate. All right, and you're going to see how cardinal it is, the ATP, the glutamate, all those things, rhodopsin, how they are able to interact and cause what we call transmission of information from chemical into electrical, then also again into actually it's supposed to be light, chemical, then electrical. Yeah, so you're going to see how that moves. And basically, when I said you need to know the anatomy before you start looking at this, because if you look at this and say, maybe for synaptic terminal, when you go back to synaptic terminal, it's saying in the outer plexiform layer, synapsing with dendrites of bipolar cells and horizontal cells. So if you went through the previous videos, I'm sure you know what bipolar cells and horizontal cells are. If you look at that diagram, it's trying to explain or point out where you can find horizontal cells and where you can find what we call bipolar cells. So these are all synapsing with what we call the light sensitive cells. So now let's look at the cones, okay? So cones are the outer segment. The outer segment is small and conical, okay? Sorry. So I don't need to go through this because we did discuss it. but just know these are rods and cone cells, and outer segment, inner segment, and synaptic ending. So they're just trying to basically differentiate between the two, the cones and the rod cells, as you know. Yeah. So now the cones outer segment, the outer segment is small and conical. Inner foldings of cell membrane from circulars. Okay. Then we have got the photopigment synthesized in the inner segment. So now when you look at the inner segment, it contains organelles with many more mitochondria. Okay. Yeah. So this one contains many more mitochondria, yes, compared to the rods. So now we have got also what we call the cell body. So from the inner segment enters the inner nuclear layer through external limiting membrane and forms the cell body that possesses the nucleus. Right. So from the inner segment enters the inner nuclear layer through the external limiting membrane and forms the cell body that possesses the nucleus. So that's it. Then when you go to a synaptic terminal, in the outer plexiform layer, synapses with the dendrites of the bipolar cells and horizontal cells, and contains the same neurotransmitter that we talked about, known as the glutamate. But how they synapse in the cones and also in the rods is a bit of a difference. If you go back to the video, we said one is 1-to-1, the other one is synapses many. Yeah. So in terms of stimulation, there's a difference. In terms of exciting a cone and the rod cells, there's a difference in terms of synapsing. All right, now let's go on the functions of the rods and the cone cells. So rods are very sensitive to light and have a low threshold. We discussed this, all right? So you don't need a lot of light in order for you to excite a single rod cells, okay? So responsible for dim light vision or night vision, or what we call scotopic vision. So the rods are responsible for this type of vision. So in case maybe a question is brought in multiple choice and they talk about scotopic vision, okay, or night vision or dim light vision, they're just basically talking about low threshold stimulation. So don't get confused about that. So do not take part in resolving the details and boundaries of objects, visual acuity, or the color of the objects or color vision. This is not a job for rod cells, okay? So vision is black, white, or in the combination of black and white, namely gray. So we usually use this type of cells when there's less light or it's in the dark. So cones have high threshold for light stimulus as compared to the rod cells. So sensitive only to bright light for bright light and daylight vision, or what we call photopic vision. So which is the opposite of scotopic vision, which is for the rods. So for acuity of vision and color vision, we use what we call the cone cells. No wonder they need more photons of light for us to be able to stimulate what we call the cone cells. So this is just a diagram showing the visual spectrum, okay? Yeah. So we said it ranges from what we are calling 350 to somewhere 750. So if we go above that or we go below that, it's difficult for us to be able to get a clear image. Okay. Now, if you can see on top there, blue cones, rods, it's black, then green cones and red cones, because there are about 3 different colors that the cones are able to perceive or view. So now, chemical basis of visual processing. What is the chemical basis of visual processing? Okay. What is the chemical basis of visual processing? That's what you're going to be looking at. Please pay attention to this, because this is the physiology that we have all been waiting for. All right. So now, ionic basis of potentials. Again, we go back to cell physiology, just like always, we'll be going back to cell back and forth. So no wonder you need to appreciate cell physiology, please, and understand it. Now, sodium channels in the outer segment of the rods and cones are open in the dark, okay? This current flows from the inner to the outer segment. So now, basically what you are seeing here, these are rod cells, if you can see even the shape. So we have here a sodium, potassium, ATP pump, okay? Then this is a pump. This makes more sense. If you look at Here we have with mitochondria. All right. So this sodium can either go that side or it can go this side. So there's dark and there's light. Okay. So we'll be basically talking about how this sodium is able to move and cause what we call either glutamate release or suppressing the production, or the release of glutamate. Basically, that's what determines either being able to see something or not. All right. So that's what that's the science, the physiology we're going to be talking about. So now, sodium channels in the outer segment of the rods and cones are open in the dark. So this current flows from the inner to the outer segment. So current also flows to the synaptic endings of the photoreceptor. So the sodium-potassium pump in the inner segment maintains what we call an ionic equilibrium. Let's go through that once more. Sodium channels in the outer segment of the rods and cones are open in the dark. So in the dark, sodium channels in the outer segment. So when you look at this, where can you find the outer segment? Is this part, okay? This is the outer segment. So sodium channels in the outer segment of the rods and cones are open in the dark. They are open in the dark, okay? Current flows from the inner to the outer segment. So this is what I'm just trying to do. Current will be flowing from the inner, will be flowing to the outer and inner segment, all right? Then current also flows to a synaptic ending of the photoreceptor. So sodium potassium pump in the inner segment maintains what we call an ionic equilibrium. So when ions are being lost, meaning sodium is being lost and potassium is being taken in. So sodium and potassium, they're being exchanged by the same pump. Why is it being called a pump? As you know, basically there's ATP being used, right? And since it's being called a pump, meaning it's going against position gradient, as you know, if you look at the way, this is the ECF and this is the ICF. So if sodium is going to the outside when there's more sodium, that is known as pumping. If potassium is going in an area where there is more, that is known also as pumping. Hence, this qualifies to be called a sodium-potassium ATPase pump. Then the release of synaptic transmitter is steady in the dark. Okay. So the release of synaptic transmitter is steady in the dark. So there's potassium there and the sodium light being shown there. So now let's go to the next slide. So when light strikes the outer segment, reactions initiated closes some of the sodium channels. So when light strikes the outer segment. So basically, as you know, the rod's light strikes the outer segment, okay, where we have got these membranes or this containing the cytoplasm. So when light strikes this area, what happens, okay? So that's what they're trying to say. When light strikes the outer segment, reactions initiated close some of the sodium channels, okay? So when the sodium channels are closed, meaning sodium stops flowing into the cell, okay? Yeah. So results in a hyperpolarization receptor potential. This reduces the release of synaptic transmitter. So if there's reduction of the flow of sodium, which is caused by light, it results in the reduction in the release of the synaptic transmitter, which is known as glutamate. So when sodium channels close, glutamate is stopped. Okay. Sodium channels close, glutamate release is reduced. Okay. So now reduced inhibition of bipolar cells increases their activity. So reduced inhibition of bipolar cells increases their activity, ultimately leads to action potentials in the ganglion cells. So the action potentials are transmitted then to the brain cells. Okay. Yeah. So when there's less glutamate being released, which is caused by closure of sodium channels, which is also caused by light hitting. So when light hits the outer segment, it results in the reduction. It results in the closure of the sodium channels, which results in the reduction of the release of glutamate, which causes the inhibition of bipolar cell, which causes reduced inhibition of bipolar cells, or which causes the bipolar cells to become more stimulated. Okay, hence ultimately leading to action potentials going to the ganglionic cells. The action potentials are then transmitted to the brain via what we call the optic nerves. So let's go even deeper and dig deeper what is happening here. So don't forget, the first step is light strikes the outer segment. If you are being confused what the outer segment is, this is the part called the outer segment. When light strikes the outer segment, we have got channels here known as sodium channels. The sodium channels are going to be closed. Once the sodium channels get closed, they're going to cause the secretion of glutamate to be suppressed or inhibited. So once the release of glutamate is reduced, it's going to cause stimulation in the bipolar cells. Then the bipolar cells are going to transmit an action potential to the ganglionic cells, which will transmit another action potential going to the brain for interpretation of the image. As you know, an image appears upside down on the fovea centralis, if you go and look at the notes for accommodation. So now let's look at this. So now, closure of the sodium channels. Please don't forget that. If you can't, if you've got any comments, please let me know so I can be able to explain more on that. So now, closure of the sodium channels. There's chemical response, okay? Yeah, we are still talking about the physiology. So what you are seeing here, please don't get confused. We are still on the rod cells. So spin, okay? So rhodopsin has got what we call, this is a rod disc membrane, then the cytoplasmic surface. This is a rhodopsin being seen here, and there's what we call intradiscal surface. We'll look at this diagram, don't be worried. So this is what causes stimulation to happen in after the closure of the sodium channel. So we'll talk about what will be happening here. So now look at the closure of the sodium channels of chemical response. So rhodopsin is a photopigment, okay? When you say rhodopsin is a photopigment, what it means is that it's sensitive to light. As a protein called opsin, okay? Opsin is a protein which is known as scotoopsin, okay? So scotoopsin is basically a protein. So there's the retinol derived from the carotenoid substance known as B-carotene, present in carrots, is present also as well there. Now, retinol, that means they're talking about this era, there's retinol here. No wonder the issue of you guys eating a lot of carrots comes from this, because it helps them to be healthy and etc. etc. So now the retinol is present in the form of what we call 11-cis retinol, known as retinine 1. Okay, so now the retinol is present in the form of what we call 11-cis retinol, known as retinine 1. Please pay attention, because the structure of this molecule is different with the one that is found in cones. So we're talking about rod cells. So the one that is going to be found in cones will be different. Yeah, so here we have got 11-cis retinol, known as retinine 1. And it's coupled to G proteins, means it's attached or combined to what we call G proteins. So now this is just a mechanism diagram trying to show the cascade or the pathways about this. Now, light has the ability to change the shape of the retinol, okay? Light has the ability to change the shape of the retinol. So let's go back. This is the structure. So light has got the ability to change that shape. So never where it's conformation or change shape, it leads to something in chemistry or in biology, or in physiology, as you know. So light has the ability to change the shape of the retinol. It converts it to all the trans isomers, all right? This alters the configuration of opsin. So once the retinol is changed, it alters what we call the opsin, which is the protein there. The opsin changes activate the associated heteroterameric G protein known as transducin. Okay, yeah. So now we'll talk about this using this diagram. So the G protein converts the GDP for GTP, and the alpha subunit separates. So now what you are seeing here, this is opsin, right? And this is 11-cis retinol that is found in the rod cells, all right? So now the outer segment membrane, as you can see there. Then we can go, this is the outer membrane segment, as you know, because we talked about the outer segment being stimulated, which is found at the front part. This is the outer segment. As you can see, that's the outer segment. Then these are the stages or steps which you can be able to follow. Then these are the sodium channels, which we are talking about. Remember the steps before we even start looking at this. And this is the inside of the cell and that side. So that ICF, ECF. Then these are known as, this is the disc, then there's a disc membrane there. Then there is a light and there's rhodopsin. So rhodopsin rhodopsin, as you can see, rhodopsin here, as you can see it. Yeah, then there's the conversion from GDP to GTP. Yeah, and then there's a protein known as transducin, as you can see. So follow this diagram step by step. So now remember what you learned in the previous slides. We said, firstly, there has to be light that has to strike in the outer segment. After that light strikes the outer segment, what's the first step that happens? There's going to be reduction in the release of glutamate. Okay. Once there's reduction in the release of glutamate, then it's going to it's going to stimulate the bipolar cells, and the bipolar cells are going to create an action potential, which is going to be transmitted to the ganglion cells, and the ganglion cells are going to send another action potential via the optic nerves. So now basically we're just talking about it happening in the outer segment, all right? Yeah, which is causing sodium and glutamate to do whatever science is there. So now we have number one. So light stimulation of rhodopsin leads to activation of what we call the G protein or transducin. So when light enters, okay, so this light which strikes rhodopsin, which is going to cause, or which is going to lead to the activation of a G protein and transducin, okay? So if you can see here, there's a G protein which is going to be activated, okay, from GDP to GTP in the presence of what we call transducin. So now, after that, we are going to go to what we call stage 2. So now at stage 2, this activated G protein activates what we call the cyclic GMP, which is known as phosphodiesterase, okay? So this is phosphodiesterase is an enzyme that is able to activate, okay? Yeah, so now activated G protein activates cyclic GMP, which is going to cyclic GMP phosphodiesterase or PDE. Okay, so this is what they're just trying to talk about what is happening there, right? So these are substances that are found on this disc membrane. Then after that, you go to DPE. Okay, this is phosphodiesterase is going to now activate, it's going to hydrolyze, it's going to act on the cyclic GMP, it's going to act on what we call the cyclic GMP, reducing its concentration. So it's the enzyme. So the first thing is that when transducing and the GDP is going to GTP, which is going to activate what we call the cyclic GMP via what we call the enzyme known as phosphodiesterase. So the enzyme has to be formed. When this enzyme is present known as phosphodiesterase, which is known as PDE, is going to now act on the cyclic GMP, reducing its concentration. So once the concentration of the cyclic GMP is reduced, so if you can see, it's going to be broken down into smaller segments. So here it's being broken down. You can see GMP being broken down, okay, by the enzyme known as PDE, which is phosphodiesterase. So once it's broken down, or reduction in GMP on the channel leads to what we call closure of the sodium channels. So light strikes to activate the conversion from GDP to GTP. Then the GTP is going to activate an enzyme known as PDE, and that PDE is going to go now, or is going to be transported to the membrane to cause or to break down the cyclic GMP into monomers. Okay, so by doing that, it's going to cause the closure of what we call the opened sodium channels. Remember, in the dark, these channels are always open. But once there's light, the channels are supposed to be closed. So in the dark, channels are always open. And when channels are open, glutamate is always being released. But when they close where there's light, okay, so in light, the channels are going to close, the sodium channels are going to close, hence they're going to suppress the production of glutamate. So once that happens, as you can see, the closed channels, once they are closed, then this leads to closure of sodium channels as GMP are being taken away from the open channels. So once that happens, then information can be passed on to what we call the bipolar cells by stimulating them. So this is basically what happens there. I hope that was easy. So this is just another version of the same story happening. You can be able to appreciate in so the alpha subunit activates the GMP. The alpha subunit activates the cyclic GMP phosphodiesterase, which converts the cyclic GMP to 5-GMP. The cyclic GMP normally acts directly on the sodium channels to maintain them in the open position. and decline in the cytoplasmic cyclic GMP concentration causes some sodium channels to close. This produces the hyperpolarization potential. So this is another diagram, another version trying to explain the same thing. Okay, so A, in darkness, rhodopsin is inactive. Okay, so cyclic GMP is higher and ion channels are open. B, light bleaches rhodopsin, opsin decreases cyclic GMP, closes sodium channels are hyperpolarization in the cell. C, in the recovery phase, retinol combines with opsin. So this is just the same thing. Yeah, another thing talk about B-carotene main source is carrots, cleavage in the body, vitamin A or retinol, bleaching by light, then the retinol known as aldehyde of vitamin A combined with lysine reduces of the protein and opsin to give a shifts base leakage. And then there's also rhodopsin. Okay, that will, yeah. So now resynthesis of rhodopsin. So this is just basically a diagram just trying to show how we can be able to resynthesize what we call rhodopsin. Okay, so it's just basically the opposite, yeah, by doing that. So you can go through this diagram in your own free time. So all trans retinol is converted into 11-cis retinol by the enzyme known as retinol isomerase. Then 11-cis retinol immediately combines with this scotopsin to form rhodopsin. So there's a role of vitamin A there. Okay, all trans retinol is converted into 11-cis retinol by activity of enzyme retinol isomerase. So it's converted into 11-cis retinol, which combines with scotopsin to form rhodopsin. All trans retinol is also converted into trans retinol. So this is just a diagram talking about how vitamin A and other things bring about the formation of what we call rhodopsin, right? Yeah. So this is just a diagram talking about the stages in which rod cells can be stimulated, starting from stage 1 up to the last stage. So now for the transduction in the console, now we are done with for transduction in the rod. So this one basically is just the same with what happens in the rod cells, with just minor differences that you're going to see in the next few slides. All right. So now cone pigments. Now there are 3 different kinds of cones. There's what we call synopsin, which is blue, which is 40 to 20 nanometers in terms of responsiveness. There's that we call idopsin, which is green, which is about 530 nanometers. Then there's also what we call porphyrospin, which is red, which has got about 560 nanometers in terms of responsiveness to light. So what this diagram is saying, so if you look at this is known as the electromagnetic spectrum. And here, this is where we have good visible light. Remember, we said under visible light, we have got 350 to 750. So it's just trying to talk about the different responsive or different colors that are found in the cone cells, as the pigments have been seen. There's blue, green, and red. Okay? And those are some of the names that are given to them. So now, only one of these pigments is present in each cone. So opsin in the cone is photopsin, which is different from scotopsin, the protein part of rhodopsin. So opsin in the cones is known as photospin. Okay. So now, which is different from scotopsin, the protein part of rhodopsin. Okay. Remember in the beginning that we talked about this, which is different from scotopsin, which is present in what we call rhodopsin. So there's here short wave, medium wave, and long wave cones, as you can see. So now, phototransduction in the cone. So light activates what we call retinine _1. Okay, yeah. There we had 11-cis retinol. Retinine. Okay, yeah. I think, yeah. So now this activates what we call the GT2, a G protein that differs somewhat with the rod transducing. So the GT2 in turn activates what we call activates what we call phosphodiesterase, catalyzing the conversion of cyclic GMP to 5-GMP. So this results in closure of sodium channels, decreasing the intracellular sodium concentration and hyperpolarization of the cone synaptic terminals. Yeah. So there's dark adaptation of light signals. So basically, it's almost the same thing. The only difference has to do with just how what is there about light activates the retinine one. So now, dark adaptation. So person is able to see objects in dim light, usually after exposure to light. After some time, eyes get adapted, adapted, objects can be seen. So after adaptation, the objects can be seen. So maximal duration for dark adaptation is about 20 minutes. So increased sensitivity of rods as a result of resynthesis of rhodopsin. In bright light, most of the pigment molecules are bleached or broken down. Time for regeneration of rhodopsin causes the delay in seeing objects. So that's what causes dark adaptation. All right. So now we're about to conclude what we started. So this is just all about the rod and the cone cells. So dark adaptation, this is a diagram just trying to show what happens during dark adaptation in rods and cone cells. Okay, so cones are the first to be desensitized. Rods maintain most visual process due to their low TH. Light adaptation so eyes get adapted to increased illumination. Discomfort is due to rapid breakdown of pigments, bright light. So we are going to end here until next time. Thank you very much.